So I'll just quickly go over this. The Earth Microbiome project was started in 2010. It was an initiative to try and coordinate a systematic characterization of microbial life. Along the same lines as the barcode of life, we use amplicon sequencing to generate a comprehensive interpretation of the microbial patterns we see between ecosystems, primarily led at the moment but by Rob Knight on the right there, Janet Jansen in the middle, and myself as a US coordination group with a collaboration of a little over a couple of hundred people around the world. Our mission is to systematically characterize microbial diversity, understanding the patterns of microbial diversity, the processes that drive these patterns, and association of different systems and scales. Obviously, the microbial problem is significantly scalar, so we're trying to understand how those dynamics are actually interpreted. Crowdsourcing is key. We don't actually go out and do any sampling. We work with the communities that have taken environmental samples from every environment on Earth, including places like this lecture theatre, to actually try and understand how we can get those samples in and process them. We're creating a framework for developing a predictive understanding of microbial dynamics. That's key. We're developing models. Models are what the EMP is all about, predictive understanding for the future. Being developed for the community, by the community is our motto, and immediate, free, and open access data. So we sequence the data, it goes public. We take the metadata, the associated physical, chemical, biological variables, it goes public. And we're providing a comparable environment. So we have standard protocols for developing and processing and producing data, which means that all data can be comparable. So anybody can take up the EMP standard protocols and develop their own study, and it will be comparable to the EMP's data sets. Our assets, we have 102 collaborators who have currently actually sent us physical samples, pledging 60,000 samples from various different groups. We processed or in the literally about to sequence around 22,000 environmental samples. 7,137 have been completed for 16S ribosomal RNA. It's a universal marker for bacterial diversity. Has its limitations, but it's the best we have at the moment. Representing around 25 environmental biomes, about 750 million sequences, and about 279,000 species currently analyzed. The public dataset is being analyzed on GitHub in a crowdsourcing effort, so anybody can get involved, anybody can help us out with this, and it's being led by Greg Caparasso. We're generating network analyses so we can understand how bacteria are shared between environments. We're working with the community to actually look at these systems with the goal to not only covering the map, but covering the environmental parameter space. So, individual environments have particular environmental parameters at particular times. That's what we're interested in covering. So, for example, here, it'd be great if we could find some more soil samples with a pH between 6.8 and 7.3. So I always say, if you have soil samples in that range, please contact us. We could help provide data for your project. In turn, you could help us provide a better characterization. Just zipping through this, the EMP has characterized about 82% of the known tree of life by systematically surveying environments across those environmental parameter scales and finding different environmental features such as alpha diversity and novel features, such as we know if we go into ocean floors and grassland soils and creeks, we'll find a lot of novel bacterial diversity. The most prevalent microbe in the EMP at the moment is limnohabitans. This is because Trina McMahon dumps tons of lake samples from Gleon on us. So that we know that we have biases in our representation of different environments, and this is key to what we're trying to do. Getting involved is done by participation. Membership is not an official thing. If you're working with us, then we're collaborating together, and that's what's key. Send us your samples. If you don't mind everything being publicly accessible and all the data being made publicly accessible, then, which you shouldn't do, then we can always work with you. And you can also help analyze data on GitHub. So we want to get the best bioinformaticians, the best data analysts, the best statisticians to work with us on this data, and that can be cool. Thank you.